Hi, so this process begins with pre-mRNA, which has a 5' cap for stability and also has exons and introns. The introns need to be cut out because they are not used for genetic coding or protein synthesis. So the way that it's done is in a complex called the spliceosome, which it contains other proteins and SNRPs. SNRPs are made up of S and RNA, which the SNRPs detect the junctions between the exons and the introns in order to slice it and cut off the intron and exit. It stays in the nucleus, but it exits the spliceosome and is degraded. And then the result of this is a new mRNA, or a mRNA where the exons are connected and there's no more intron, and it also has a poly A tail, which is there for stability and also for the new mRNA to exit the cell through the nuclear pores.
Go.
Okay, so in this process you have activators which bind to the distal control elements, which are grouped together as the enhancers, and DNA bending protein will cause the strand of DNA to fold over and get the activators closer to the promoter, which includes the TATA box. So the activators will bind to mediator proteins and general transcription factors, which will create an active transport initiation complex on the promoter and produce RNA synthesis, which is really important because without this process, very little RNA would be produced.
3.
Okay, so in this figure we have two different scenarios. In the first one, there's no lactose present. So you have the LAC1 gene in the LAC operon, which codes for the active repressor. The active repressor is by default active and attaches to the operator. And in that way, the RNA polymerase is unable to go down the chain and express the gene. But in the second scenario, we do have lactose present, and allolactose attaches to the repressor, making it inactive and unable to attach to the operator. So the RNA polymerase will be able to go down the chain and express the different genes. The different genes we have are so the genes that are expressed are beta-galactosidase, which changes lactose to galactose and to glucose, permease, which allows lactose to enter the cell, and transacetylase, which we're actually not sure exactly what it does. Okay, so the regulatory gene codes for the inactive repressor, and the inactive repressor is unable to attach to the operator, making the RNA polymerase possible, so the genes of operon are able to be expressed. The genes of operon code for the polypeptide subunits, and this makes enzymes which are used for for making tryptophan. In this scenario, tryptophan is already available and binds to the repressor the inactive repressor to activate it so that it attaches to the operator. And in this case, the RNA polymerase is unable to go down the chain and no RNA is made.